Hi guys! So today we're going to take a look at a paper called Drench Application of Fish-Derived Protein Hydrolysates Affects Lettuce Growth, Chlorophyll Content, and Gas Exchange by Chinping Zhu and Beiquan Mao. So first thing we need to talk about with this are biostimulants. Biostimulants are compounds that are, or things that are added to plants in small amounts to enhance the growth. The reason people have such an interest in biostimulants right now is under our current system there's a lot of questions about environmental impacts of fertilizers. The thought is if you can apply a small amount of biostimulant, you can either replace part of or all of the fertilizers you're using in order to get the larger growth you want. There's also questions on some papers have raised as far as a decrease in nutrition under our current conventional system, and this might help address that problem. For this paper, the authors used fish-derived protein hydrolysates, which are a type of biostimulant, and they wanted to look at how those affected the growth of lettuce. Now, past papers have used hydrolysates and kind of looked at them, but they were either or foliar application, so it wasn't a drench applied to the soil, it was sprayed on just the foliage. They tend to be hydroponic or soilless, so they were using either perlite or they were using sand in place of a soil. The thing with that is you're not getting any microbe interaction. The good side of doing that means you can actually get to the roots whole and clean and measure them. But like I said, there's no microbe interaction. The authors of this paper wanted to know whether that affects it, because in previous papers they only saw an effect of this particular biostimulant when it was applied during stress conditions, a lot of salt, drought, overwatering, things like that. So what they did was they took and they sowed seeds inside of just your usual soilless media they would use for planting, and then when those seeds were 2 weeks old, they transplanted them into 6-inch pots, which they filled with field soil. So they literally went out in the field, dug up soil, and put those in the pots. So it contains everything that you would have in the ground as far as soil interactions go. These plants were watered to field capacity with a nutrient solution. They also took the weight of that because they used that later to determine watering. Half of the plants were without anything else, just the nutrient solution, and the other half had those hydrolysates added to them at the recommended dosages. There were 8 pots per treatment, so 16 pots total, and they were grown up in a growth chamber to reduce any interaction with environmental conditions. The pots were rotated and irrigated weekly. When they were watered, what they used was that field capacity weight again to keep all the pots the same amount of water present in the soil. It helps reduce any of the interaction of a pot having less soil than others or whatnot, so they all have the same available water. The protein hydrolysate was added at day 0, 14, and day 24 per recommendations of the manufacturer. At the 30-day post-transplant mark, 2 of the largest leaves from each plant were tested for their gas exchange and chlorophyll fluorescence. This let them know the photosynthetic rate, the transpiration rate, and the stomatal conductance. Cores were taken from 4 leaves, and they measured those for chlorophyll, specific leaf area, and the water content. And then the plants themselves were harvested. They counted the number of leaves, they looked at stem diameter, they took the shoots and got a fresh weight and a dry weight, and they took a dry weight from the roots. So all size indicators. How big is this plant growing? As results go, they found that there was an increase in growth on all the factors. There were more leaves, a bigger stem diameter, larger roots, larger leaf area present on all the plants that were treated with the protein hydrolysate. They also found there was an increase in chlorophyll present in the leaves, and a lot more. There was a higher water content and a better gas exchange going on inside of the plants that were treated with the hydrolysates. So what they're looking at was this is probably a nutrient uptake thing. The microbes present in the soil had an effect on this most likely, as well as the root size. The root size being larger, they can take up more nutrients. They also think that there was an increase in metabolic enzyme activity related to these hydrolysates and probably the microbes as well. Overall, it was a really good study. They took out a lot of the variants and found pretty interesting results.